just put your drop down on the thing, and then you actually tilt your slide up to it, and you just kind of let it go, and then you squish it. Right. The other way, if that doesn't work very well, you can get a paper towel, and you can just basically drag it from one end to the other, and I'll show you that. One way you can do this is another way. People do this with blood all the time. They'll put a drop on the side, and some capillary action will pull it in. And usually they have another liquid under there, like a buffer or something like that. And then when they put the cloth right here, it ends up pulling the liquid through by capillary action. So that's one way to load a cover slip as well. So sometimes people will take these cover slips and they'll put petroleum jelly on their palm, and they'll just end up scraping it off on the edges. And they'll just go around the cover slip and scrape off some petroleum jelly. And then what they'll do is they'll flatten that petroleum jelly on that drop, and that will create a seal around that wet mount. So they'll put it like it's a little dry chamber, a little wet chamber in there, and that allows the cover slip to be held in place. So when you're done with this, you can take clear nail polish, you can put around the edges, and that will seal in any liquid so it can't dry underneath there. Okay, so that's one way you can often do that. So yeah, it's nice. But you sure do get the weird looks when you're buying at the store. Well, you might not get weird looks. I get weird looks. This won't stay. This nail polish is pretty old, so I don't think it's going to seal there. Okay, so if you guys look at these guys, see how dirty they are?
Yeah.
Okay. Again, they're both 10x. Okay. Independently focusable, I think.
How do you clean them?
I'm going to show you that in a second. But actually, should be.
Maybe they need lubricators.
Yeah, they're a little old. I don't want to mess with them, but
Okay.
they should be movable, and they are off. So, nose bridge adjustment. Okay, no big deal. If you look at the objectives too, you see someone tried to clean it with soap. So you can see residue on there because of the soap, and it kind of looks like bubbly. This should be cleaned as well. This is okay. It's using chem wipes for the lenses, which is no problem. Chem wipes actually, they claim to be lint-free, but they're not. Chem wipes actually do have a lot of lint on them.
But do they damage the lens?
And they can scratch the lenses.
Yeah, it's pretty hard.
So, yeah.
You get lens paper?
We do. We have lens paper.
Oh, okay.
You can actually get Q-tips too that are like lens paper-ish Q-tips.
We need Q-tips?
No, they're lens paper grade Q-tips, so they're really expensive. Okay. So for this one, it's a little bit different. Well, let me teach you how to clean it first. Then we'll get an operation. Okay, so this is just a cleaning kit. So it has cleaning solution in it, and it also has lens paper in it. The cleaning solution is a mixture of alcohol and some mild soap, so some mild detergents. So it really isn't that bad. For things like this, you can take these guys out and you just put a drop on there. Some people put drops on the actual paper. But for this, you don't really want to wipe. You just want to kind of rub in the middle. It's not like, I'm not really putting any pressure there. I'm trying to get most of that off. But most of this should actually evaporate by itself. So you shouldn't have to worry about it. And that should get at it. The motion, you don't rub. Like, it's not this way, it's circular, and you start from the inside and you go out to the edges, because realistically the very edge of it you can't really clean very well, so you want to push all the dirt to the edges. That's what you'll do. So That's it. So we'll start with the other one. You don't really need that much. That's it. So one other thing that Zeiss recommends too is actually breathing on it. By doing that, you actually put distilled water on it from your breath. Now, people claim, yeah, you'll pick up microbes from your breath as well, so it's just easier to use cleaning solution. That's it.
They'll be at 1 power and not at 400 power, so it won't matter. At least the ones on the top lens.
Yeah, but they'll create residue, and if
Yeah.
you get fungus on your objectives, they can eat the coating away.
Oh,
so you don't
okay.
want to put anything on there like fungus or anything. So that's kind of, you can do it. Zeiss actually has it in the little cleaning microscope book here somewhere. I was like, I don't think I want to do that.
So you don't clean the inside.
The inside theoretically should be clean.
Yeah.
Theoretically. Yeah. There's fungus. So every one of these guys should be able to come off. Usually if you end up taking one off, you put a piece of tape over that.
Yeah.
Okay. I just want to clean this. Some people, you don't have to film all of the cleaning. It's all the same thing. Okay. This is the stage, so you can move the stage around. Okay, and typically the slides will just sit on the stage. So you just put it in there and then just zoom it around to wherever you need it to be. Okay. Let's see some of the other features in the microscope. So that's basically the bottom. This is, if we have the light source here, this is to focus the bottom light source. And usually it's a condenser unit that sits here, and then the light source is down here, so that condenses light into the top. So, and usually the same thing, you'll have a diaphragm, and you'll be able to choose, you know, what sort of you roll it down to. So, and now I'm going to show you how to line up the actual light source. And when I came in, this was one of the first things I wanted to check, because I need to know how to take this thing apart, so I don't look like an idiot when I actually end up turning it back on. So, okay, so that is the actual filament in the lamp.
Okay.
When you come in, it usually looks like that, right? So you want this to look like this. Okay, you want it to be adjusted to be like that. What's that?
What's the purpose of getting the filament in focus there? Is that?
That will align the wavelength of light, so you'll get everything. Because when things are kind of out of focus, then it's not very helpful. You want things to kind of be in focus like that. Because we'll end up putting it back in here. So let me get my thing in it. Let's step through this stepwise. I'm gonna go grab that poster because it percent in, and it's 50 to this and 50 to this. So,
okay.
but once we change this out, I want to look inside here and see if there's a dichroic. Because if there's a dichroic, then we can start doing some really interesting stuff. But I don't know until we get that done. So, and I'll explain what I work on too, which is a little bit different. It's so let's just say green. Okay, so the first filter selects green out. Okay, all the rest of the colors don't go through this first filter. Your dichroic is selectively reflective to this color. Okay, and it will pass that color up to your sample, and your sample will fluorescent. Will absorb that color, and it will fluoresce in a different color. So it changes the wavelength of the color it emits.
So it may glow yellow or purple or something.
Usually it glows at a higher wavelength. So if it absorbs blue, it emits green. If
that makes sense.
If it absorbs blue, then it emits orange and goes on from there, right?
Is that based on the dye that you've treated the sample with, or?
Yes, it's based on the dye.
Okay.
Okay.
So the color of the dye and the dye that
So
what
you
it
can
absorbs.
you can specifically
Right,
use
you
a
can
dye.
tune-
You can tune the dye to what. You,
Right, and some of them literally it is tuning, like quantum dots. They are tuned by their size, by what they'll emit. So the bigger the size, the darker the color. Like darker, meaning it goes up in wavelength to
towards red.
towards red.
Yeah,
So it'll go
okay.
blue, green, red. But they all quantum dots just absorb mostly in UV, so and emit invisible. So
Is quantum dot a class of dye then?
quantum dot is a nanocrystal. It's a semiconductor. It's a cadmium selenium crystal. So they're really cool molecules. But traditional organic dyes, fluorescent dyes, they'll absorb different colors and emit, and it all depends on their just emission properties.
Right. I see. Okay.
So anyway, so this blue light will actually end up entering, right, and it's really faint. How you doing? It will enter and it will pass through your dichroic. Okay, where this green light that's reflected off the surfaces, right? Maybe there's a dust particle, and that green light ends up coming back. That green light is reflected back,
I see.
right? Where the blue passes through.
Right.
Okay. So the blue, there's all sorts of light coming back too, as well. There's reflections off the inside of this. There's reflections off this. There's reflections in here. So they have another filter here to clean this up. Okay, and it's called the emission filter. So this selects the wavelength you want. Okay, so it's an additional filter in case some of this gets through as well. Right, that bounces off. Okay, then it goes to your detector, whatever your detector is. So if your detector is a camera, then it goes to that. If the detector is, your eye, then it goes to your eye. So, so it just depends what it does. But that's basically like dichroic and
Cool. That's great.
fluorescent theory.
What magnifications do you typically run in this kind of microscope?
Typically it's 100x, so it'll be 1000x. So
1000x.
right.
Oh, okay.
So
you're
usually
looking at small.
Right. You're usually looking at cells or certain
Internal
small
structures
things.
of
Yeah,
cells.
yeah.
Yeah.
So you can see they use some of these dyes in internal stains in eukaryotic cells. So they'll aggregate around organelles, or they'll tag the mitotic juncture. They'll tag specific places in the cell. They'll mark it, and then we can see it. Mitotic spindle. They
Like
tag
the mitochondria, or
yeah, they can tag the mitochondria. They can tag the Golgi apparatus, whatever they want.
I see.
They can tag specific small molecules and see them traffic in the cell. So they can see the movement from one side of the cell to the other. They can see intake of molecules as well. So they can see it being pulled into the cell.
Is the cell alive after it's tagged and everything?
Yeah. The cells, some of them can live with the dyes in there. And
Cool.
like GFP is made by the cell, so they can tell when a protein is getting made by the cell because it's tagged with GFP. So when you put a GFP marker,
And it'll
on a
fluoresce like
gene,
this.
when that product is being made, like our GFP cells here,
Right.
you can see, when you put a arabinose on the slide, you can see just a few minutes later that they turn green. Then you can see where it comes from in the cell as well, where it turns green. So, yeah, it's really interesting.
So you typically have like a large selection of these kinds of dichroic filters and other filters in order to get the effects you want.
Typically, you'll have 4. I'll point this out later, but typically you'll have 4 sort of filters.
Okay.
Here's a dichroic.
Yeah. Well,
It's very selective.
I don't remember what, and it reflects. Oh, it reflects green and it passes red.
So if you have a light source through there, you're going to get some red through it, but the green gets reflected.
Yeah, because you can see the reflection on there, right?
Oh, yeah.
It's reflecting green back
absolutely.
to us.
Yeah.
But we can look
So
through
it's
it.
red on one side and green on the other?
But if you look through it, you're going to see red. So.
Yeah, exactly.
Right. That's cool.
And then, should I be seeing something different on this side?
You might be able to.